Alright then, this is the second part of the review for lipids. First part we finished covering saponifiable lipids, and I said that these sphingolipids actually belong down here. So sphingolipids, they are non-saponifiable, meaning there's no ester. And actually, at least one variety does have a phosphate ester, or phosphoester, but not all of them do. And they certainly don't have a carboxy ester. So sphingosine, if you remember, it's this long tail here, and then there's three things sticking off. Now one is an alcohol group that isn't used. Right. One is an amine, and then there's another alcohol here in the bottom. So you don't really need to know the whole structure, but this business end. This alcohol group is not used for anything, but the amine and the other alcohol here at the bottom are. So in a cartoon form, we have our sphingosine backbone, and to this nitrogen here we will condense a fatty acid. And this bond between the fatty acid, would be the carbox, or the carbonyl, the carboxyl of the fatty acid joined to the nitrogen here. That's not an ester, this is an amide. Amides are not saponifiable. The other alcohol we have down here, this is esterified in a phosphoester. And a phosphoester to an amino alcohol. So these are the same amino alcohols that we saw in the phosphoglycerides. Generally it's choline or ethanolamine. So choline or ethanolamine. And if it's choline, this is referred to as sphingomyelin. The nature of this amino alcohol here is choline and sphingomyelin. This forms the myelin sheath that protects nerve cells. We could also have, instead of this phosphate and this amino alcohol, so let me draw another one of these guys. Won't bother with the structure, but again, it's a amide bond. We have a fatty acid, an amide bond here, and here we could have a sugar molecule attached to it. Now this was an alcohol, remember, right? And the sugar is attached in a glycosidic bond, which is an acetal bond. An acetal or glycosidic bond. Now if there's only one sugar, okay, we call it a ceramide. These are also found in nervous tissue. If there's two or more, generally there's not thousands like you'd find in a starch, but it is, you can have 4 or 5. So I'll just say more than one sugar. We have a ganglioside, also found in nervous tissue. So you think of this as a gang O sugars. We have a number of sugars attached here in the glycosidic bond. Alright, steroids. Cholesterol, bile salts, hormones. Steroids have this cholesterol nucleus, and I don't expect you to be able to draw the steroid nucleus, but it looks something like this. And there's another. Something like that. So there's 4 rings in the steroid nucleus. Actually, that's not right. Curves I get there. 1, 2, 3, 4. 1, 2, 3, 4, 5. Yeah, that's right. Okay. Oop. Here and the cyclopentane there. Okay, so there's we go. So cyclohexane, 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 cyclopentane all fused together, and there's an alcohol down here. Alcohol is what makes this thing end in OL. Cholesterols, they're alcohol, they end in OL. And there's another, there's a alkyl chain that comes off here. You don't worry about the nature of that. But this is the only compound that we talked about that has that looks anything close to this. All right. So cholesterol is found in a number of things. Well, it has a role in cell membranes. It regulates membrane fluidity. The more cholesterol you have in the membrane, the more rigid it is. Now, one thing you should know about cholesterol, I'm just talking about cholesterol now. Cholesterol it's in the diet, but it's not necessary to have in the diet. It can be made in our cells, so not necessary for our diet. Can be made by our cells. Cholesterol is transported in the blood not as free cholesterol. Right? Cholesterol is very hydrophobic. It would not be soluble in water, but instead it is transported in those lipoproteins. So it's transported in blood via lipoproteins. For example, LDL and HDL. Can also be esterified. You can have fatty acids esterified to them. Can be esterified. So even though under here steroids, these cholesterol, bile salts, hormones are called non-saponifiable, there is one type which can be saponifiable. That's a cholesterol ester. Can be esterified to a fatty acid. Bile salts. This alkyl chain here ends up getting a carboxyl group, essentially an acid. So bile salts are salts of the acid form of cholesterol. And we talked about how soaps are the salts of fatty acids. Well, this is the salt of a cholesterol acid. So there would be an acid group up here. Let me just COOH. Right, so there would be an acid group. And if we make the salt of this, we have this long hydrophobic tail and a polar head group. It behaves just like a soap. Behaves like a soap. In the lab we talked about emulsification and emulsifying agents. So these emulsify lipids in our diet. simply aid in digestion. So cholesterol, bile, lastly hormones. The hormones, if you recall, there are two types. There are the sex hormones, formed in the reproductive glands, the ovaries and the testes, and they are naturally involved in reproduction and secondary sex characteristics, principally the male sex hormone testosterone from the testes, and androgen or estrogen, not androgen, I'm sorry, androgen is actually another name of testosterone. I meant to say estrogen, female sex hormone. And then also we had they call them adrenal corticoid steroids. Both sex hormones and adrenal corticoid hormones are steroid hormones. They have this, what makes them a steroid hormone, they have this cholesterol or steroid nucleus, these four fused rings. So they're produced from the adrenal glands. Adrenal glands adjacent or on top of the kidneys, adjacent to the kidneys. You don't have to worry about, just like I'm not going to ask you about the whole shape of cholesterol, I don't expect you to be able to look at one of these hormones and be able to tell testosterone from estrogen, or be able to tell cortisone from aldosterone. So the two that we talked about were cortisone and aldosterone. As you might suspect, being adjacent to the kidneys, these are involved in fluid balance. As are your kidneys, are involved in maintaining fluid balance. Also involved in inflammation. Also fever. Prostaglandins. These guys are derived from arachidonic acid. And again, you don't really need to know the structure of arachidonic acid, but know that it is 20 carbons long, and I will attempt to draw the structure of arachidonic acid, if I can remember this right. Double bond here, 3, 4, double bond, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19. I don't know if I counted that right, but something close to that. Okay, so it has this shape, usually drawn in this fashion, so where it kind of folds back on itself. Remember, these are cis double bonds. They produce kinks in the molecule. And the prostaglandins, we've said these are hormone-like because they're excreted into the blood, and they have their effect throughout the body. It's like hormones. However, hormones come from a specific gland, maybe the testes or the adrenal glands, or growth hormone from your pituitary gland or something like that, or insulin from your pancreas. They come from a specific gland or tissue type. Prostaglandins, although the name suggests that they were from the prostate gland, and that's where they're originally from, turns out they're not just from the prostate. They are found in most tissues, so not just from the prostate gland. in the prostate gland, but found in most cell types or tissue types. And so for that reason, they're not really hormones, but they do have hormone-like actions. They're involved in clotting, inflammation, fever, and also birth and delivery. Now, these three, clotting, inflammation, fever, you might be familiar with the fact that aspirin increases one's clotting time, so people who have heart disease may take aspirin a day as a blood thinner and prevent clotting. If you have a headache, maybe you have blood vessels in your brain that are inflamed, and so you take a headache to reduce that pain. And if you have a fever, maybe you take aspirin or some other non-steroidal anti-inflammatory to reduce that fever. So the fact that prostaglandins are involved in these three is also the same reason we take NSAIDs, non-steroidal anti-inflammatory drugs. So remember, we said cortisone is also involved in inflammation, but these non-steroidal anti-inflammatory drugs, such as aspirin or ibuprofen, such as aspirin, which we made in the lab, acetylsalicylic acid, ibuprofen, naproxen, acetaminophen, and so forth, have their effect on the production of these prostaglandins. So these guys inhibit prostaglandin synthesis. So if there's no prostaglandins to clot your blood, your clotting times increase and it's a blood thinner. If there's no prostaglandins to induce fever or inflammation, then these drugs will inhibit the inflammation, reduce the fever. Okay, name the major classes of steroids and their general function. We kind of just talked about that, so I'm not going to bother with that, but I just mentioned, I won't go through their functions, but we have the sex hormones and the adrenal corticoid hormones. Describe the general function of prostaglandins. I already talked about that. They're hormone-like. But they do not come from a specific gland, so they're not true hormones. You should know these other terms. Hydrophilic, water-loving, a micelle, right? A micelle, you can draw yourself a picture if that helps. Got the polar head groups of the fatty acids here, and their little hydrophobic tails going on the inside. So there's a fatty acid micelle. Lecithin. Lecithin is a phosphoglyceride that contains choline, so it's a phosphatidyl choline. By the way, it's also found in a lot of foods and in non-stick cooking spray. Soaps are the salts of fatty acids. We just got done talking about cerebrosides, hydrophobic hormone, saponification, right? This is the base hydrolysis. Base hydrolysis of esters. Also, you can think of it as soap manufacturing or soap making, the soap making reaction. And this produces the salt of a fatty acid. Glycolipids, like the gangliosides, the cerebrosides, these are the ones that, sphingolipids that had cholesterol on them, or not cholesterol, carbohydrate on them. Fluid mosaic model. All right, well, we'll talk about that down here when we talk about the composition of the lipid bilayer. Okay, for the lipid bilayer, I would refer you to the picture in the text. Picture's worth 1,000 words, but the thing to remember here the cell has a plasma membrane or a cell membrane, and these are not fatty acids. These are phosphoglycerides. I'm not doing a very good job of this, but remember, phosphoglycerides have two fatty acids attached to them. So for each polar head group, we have two tails. Right? So it's composed of phosphoglycerides we have the polar water-loving polar head groups on the outside, right? The hydrophobic tails on the inside here. This separates the outside of the cell from the inside of the cell. But we could also have proteins in here. Right, so I can use a different color for protein. Right. And you could also have that cholesterol business. Not doing a very good job of this, but remember it's 4 rings, put the alcohol group there, so you can have cholesterol in here as well. Okay, so again, here's our lipid bilayer formed from 2 layers of phosphoglycerides. And you can also have proteins like we talked about that are kind of sitting on the outside of the cell, and they may have some fatty acid stuck to them, right? So it would be a lipoprotein, and on that lipoprotein we might have some carbohydrate decorating them. Right? So maybe it's not only a lipoprotein, but it's a glycosylated protein as well. Right? And all these different pieces come together to form this fluid mosaic model. Describe the role of cholesterol. I mentioned that earlier. Essentially, the increase in cholesterol in the lipid bilayer makes it stiffer, makes it more rigid, less fluid. Makes bilayer less fluid. Okay. Alright, so that does it for this review. There are many things we talked about in lecture that were not covered here, at least not as much detail, so be sure to look over your notes and read the chapter.